Thank you, everyone, for coming to the uh, Raymond Adams Lecture. It's a real pleasure to introduce Craig Blackstone, who's the head of our movement group here at Mass General. Craig received his MD and PhD degrees from Hopkins University, and then he saw the light and moved to Boston, did his residency and fellowship and postdoc here in the system. He then joined the NIH and was there for about 2 decades, rising to the level of senior investigator and cell biology section chief. And then we were able to very happily recruit him back to Mass General, where he is pursuing both basic science and clinical activities, of course. And it's so wonderful to have you. And I think, you know, as those of us who knew Dr. Adams know well, this merger of biology and science, clinical medicine is close to all the things that he championed for so many decades. And I can't think of a better recipient than you of both this award and also of the honor of giving this lecture. So Craig, let me first present to you there.
Thank you very much. Thank you. Well, thank you, Brad. That was really a kind and wonderful introduction. And as you can all imagine, this is a great honor for me to be able to present this lecture. Before I get going, I just wanted to get some things out of the way. I have a few disclosures, which are also listed, I think, on the website. But I really did want to, as I think, follow up on what Brad just mentioned about Ray Adams. And before giving this lecture, of course, I looked a little bit about him. And, you know, as we know, he's a spectacular neurologist, one of the leaders in the whole field, as well as a neuropathologist. But as Brad said, one of the things that I hadn't really thought quite as much about until reading more was really that he was the pioneer of the science of neurology. And I think in many ways, even though the techniques we use and a lot of the approaches we use are different, in many ways, we still try to live up to that, to really understand the science of the diseases that our patients have and use that insight to create better therapies. And again, I think that Ray Adams was certainly the pioneer in that. And now thinking many years before that, another very famous biologist and clinician, Rudolf Virchow, made the statement that all diseases are disturbances at the cellular level. And this has had a lot of influence on me because a lot of the studies that I do are really trying to understand disease at a very fundamental level and to use those fundamental insights to create therapies or to, you know, sort of target the disease right at its origin and hopefully be able to impact disease before it affects people to a great degree, to modify the disease rather than treat the symptoms. Now, the disease I'm going to talk to you today about is hereditary spastic paraplegia. And many of you, as neurologists, would know what this is just from the name. And of course, even though those of you who aren't as familiar with the disease, hereditary spastic paraplegia, know what spastic paraplegia is, because it's a disease of very long axons. And those are the ones that start, as you can see in the schematic diagram here, on the right, in motor cortex, going all the way down and synapsing at various levels to lower motor neurons, usually through an interneuron. And as many of you can see, some of these neurons can be very long. They can extend up to 1 meter in length. And of course, that creates the ability for us to signal very rapidly, but it also creates a vulnerability, and that you can interrupt these tracks at many different levels. And you can imagine that the most common manifestation would be your legs, because those are the ones that are the longest. So one feature of this disease is that it causes paraplegia. It is a length-dependent disorder that affects the legs predominantly in an ascending manner. Now, there's also other forms of this. That's the pure form. There are complicated forms that have other signs and symptoms. So for many years, a lot of the description of hereditary spastic paraplegia has been based on the symptoms that we see. Now that changed particularly in the 90s and then through the 2000s, and especially with the advent of next generation sequencing. I've been working on this for about 20 years, and I think when I first started, there was like 15 genes or so, and now we're up probably to about 100 different genes. And of course, that's been because of the advances in sequencing that's made it cheaper as well as far more powerful. Now, the prevalence of this disease is somewhere between 2 and 9 out of 100,000, depending upon the population that you're in. And as I mentioned, one of the unique features of this disorder, even though it's relatively rare, is the number of different genetic loci. So you can think about it as SPG 1 to 88, but there's also others, just genes that were named for something else, but turns out that hereditary spastic paraplegia is the predominant symptom. And of those, we have at least 70 gene products. And what that gives us is it gives us the opportunity to take those genes and to begin to understand the fundamental biology. Now, at an early stage, we started thinking, well, it's not just one disease or not just 90 different diseases, that we can probably parse these into different categories. And of course, that was done for many years clinically. And of course, now we can do it genetically as well. But again, for those of you that don't see a lot of hereditary spastic paraplegia patients, I just wanted to give a little bit of a clinical outline of the disease before I launch into a lot more discussions of the pathology that we study. Now, the differential diagnosis of HSP includes all the diseases I've listed here, and I won't go down through them. I think you can see that there's a lot of different things, and you can imagine that there's a lot of ways you can alter the corticospinal tracts because they're so long, because there's so many ways that different diseases or injuries can impact them. So that as you know, many of you are familiar with seeing spastic paraplegia for many different causes, even if it's not HSP. Now, one thing I would mention about the HSPs that distinguishes them from many other disorders is they're often symmetric, they're slowly progressive, they often have distal vibratory sensory loss. Those are long sensory tracts, urinary symptoms. And one thing I do want to emphasize is that even though we have 90 different genes, about 30% of the patients that we really believe clinically have this still don't test positive. So there's certainly a big gap that we're trying to fill in terms of diagnosis. And a recent thing that we've been doing with the Spastic Paraplegia Foundation is we've created 9 centers of excellence throughout the United States, and Mass General is one, as well as Boston Children's. So we have 2 within Boston that are really and one of our real goals is going to be to try to fill that diagnostic gap, because we think that'll be very important for patients to know what the cause of their disease is, and in terms of how we'll treat it in the future. The other disease I would emphasize on this list is cerebral palsy, because we're finding that, and a lot of work is being done at Boston Children's by Darius Ibrahimi Fakhari and others, is that a lot of kids that are shuttled into these CP clinics are turning out to have HSP. So it's an important thing to remember that a lot of, when you ever ask about histories, sometimes you'll hear a history of cerebral palsy, or in older relatives, multiple sclerosis, when that diagnosis wasn't made quite as carefully. So often if you ask if you have a history of HSP, some people won't know, but then every once in a while you'll hear that somebody had cerebral palsy, MS or something. So of course that was just how it was diagnosed, but that was probably the HSP as well. So those are really the main things. I would also emphasize at the very bottom, there's other genetic disorders that can overlap with HSP. There's spastic ataxias, there's cognitive disorders. So to some degree, it's somewhat arbitrary how we assign a disease, an HS or a gene, an HSP locus, which we call SPG for spastic gait. However, over time, we know there's some overlap, but for the most part, all of these different genes have really sort of fit into nice cellular schemes that allow us to, I think, approach these disorders therapeutically. Now, when we think of treatments, the first thing you notice when you look at this list is that they're all symptomatic. With physical therapy, stretching, we try to decrease the spasticity with medications, oral mostly, sometimes even baclofen pumps that go right into your spinal fluid to avoid some of the side effects of baclofen. Pain relief, because sometimes the spasms or the stiffness can be painful. Occasionally, some of our patients have other symptoms like dystonia. A lot of them have spastic bladder. Again, we try to speak to spasms in multiple ways. Occasionally, patients will have Parkinsonism, and we treat that with levodopa. And then there's a number of off-label of drugs that we use as well. So what you can see from this list is that there's a huge need to have specific therapies for these disorders. Again, when I showed you that number 2 to 9 out of 100,000, you're really talking of tens of thousands of people in the United States. And again, this is a disorder that's underdiagnosed, so the numbers are going to be higher. So that's sort of what I wanted to talk about clinically about the HSPs. There's this great need where we've understood a lot more about it because we understand the genetics so well. But our real challenge now is to take this genetic understanding and really translate that into ideas for therapy for patients. So one of the early ways we did this, and some of these annual reviews are really old. I'm writing a new one now, so I'm hopefully thinking about how to present this, but we tried to just put the genes into different categories based on where they are within a neuron. So as you can see throughout this list, we have we have genes listed under different organelles or under different mechanisms. The advantage of this is actually it's turned out to be very useful because it's helped us sort of focus our research efforts in certain areas. The downside, of course, is that these are artificial silos, and that, of course, there's interaction between them. And in some talks, I actually focus on the interactions. In this one, I'm just going to focus on one organelle, and that's the ER. And the reason I'm doing this is just thinking about a lot of the advances that we've made in neurology over the years based on Ray Adams and others, use of pathology, is I'm going to talk about the pathology of the ER. And we think of pathology, we think of brain sections and cutting sections and things like that, and looking at them under microscopes. And so what I'll show you is that when we cut sections, we can't use blades because we cut them so thin. And when we use microscopes, we don't need eyepieces because we're working well beyond the diffraction limit of lenses. So it's really pathology, but it's a pathology in a very different way. And the reason we focus on the ER is in many ways that's the hardest organelle to image, and it's the hardest really to understand in terms of functions because it takes up so much of the cell, has so many different morphologies. And functions, of course. But as you'll see, we're starting to get enough of an understanding of the pathology of the ER to, I think, make really significant inroads into treating these disorders. The other reason to focus on this tubular ER is that the most common genes, the three most common forms of spastic paraplegia, which is about 40% or 50% of all the patients, have mutations in one of these genes. So from a patient point of view, it's an important area to focus. I would emphasize that if you look at disorders like I have on the far right, lysosomes, endosomes, autophagosomes, a lot of those disorders are autosomal recessive. And in fact, there's a lot of gene therapy work that's being pioneered in those. And those are also the pathways that overlap with some of the more common neurodegenerative disorders like Parkinson's disease. Now, if you think about what the ER looks like, this is an image from, I think, an article we wrote about 10 years ago. And if you don't remember it, it's okay because it's not correct. I mean, this is what you'll see in all the books, but what you'll notice when I show you later is it's only the surface of what the ER looks like, the types of structures that the ER has, and what its functions are. As you can see, we have ER cisternae that have ribosomes, they make proteins. We have tubes that go throughout the cell. Actually, everywhere you can fit cytoplasm in a cell, you'll find an ER tubule, and there's sheets and cisterns that store calcium and so on. But you'll see that as I show that there's a lot more structural heterogeneity to the ER as our ability to study the pathology has gotten better. Another reason to focus on the shape of the ER is not just this sort of form follows function general concept, but it's that all the genes that I mentioned have specific signatures, or all the ones that are associated with ER morphology. So if you think of all the genes that are implicated in ER shaping, there's already, I think there's 6 listed here, and we have several more now. So you'll notice that they have a common feature. They only go halfway through the membrane. And what happens when you have a lot of proteins that go halfway through the membrane and you put them together is it bends the membrane. So that's how you form an ER tubule. So we've learned a lot from just the biology of the ER from this disease. And in fact, some of the key proteins in forming the ER and some of our understanding of how this organelle is formed were from studies of spastic paraplegia. So if we take this insight and we sort of meld it or mold it onto what we already thought about the ER, we can actually get a reasonable understanding of how you create this polygonal structure of tubes with occasional sheets. And you'll see in like panel A, you can see what we typically would think of the ER looks like. In panel B, you could say, okay, how do we distribute it? Well, it's along microtubules. So microtubules are, of course, important. In panel C, you'll see that there's these proteins called reticulons, which bend the membrane, and proteins and GTPases called elastins, which actually fuse the tubes together. And the elastins were actually identified first as a HSP protein. So our whole understanding of how the tubes link to one another is from this disease. And then if we think about sheets, well, at the end of a sheet, you would bend it. So you have these reticulon proteins, and then there's other proteins throughout the sheet that actually maintain that structure. So it allows us to understand a little bit more about how cells create the morphology of the ER. And that, of course, is important. But there's a lot more to the ER than just its shape. However, one of our earliest thoughts where we were thinking, okay, as we understand the shape, we can understand the function. But early on, we realized that we had a problem. And that was that imaging for the ER is not that good. If you look at most images, they look something like this. And it's really difficult when you look at this under a confocal microscope to really understand the variation in shapes, how disease might impact it, and so on. So early on, it's well over 10 years ago, we started working, and I've listed them as ER pathologists, but my student, Johnny Nixon-Abel, we started a long-term collaboration with Eric Betzig, Jennifer Lippincott-Schwartz, and Harold Hess. And Jennifer was my neighbor at the NIH when I was there. And then she moved to Howard Hughes Medical Institute at Genelia to work with Eric and Harold. And they've really helped us, I think, understand how disease impacts the ER and how we can use those insights as therapy. And none of them are physicians, but I really do think they're pathologists. And in a way, what they've done is they've created the advances that we need. So Eric is a Nobel Prize winner, but his Nobel Prize is not in medicine, it's in chemistry. So he created a lot of the technologies that we need to overcome the diffraction limit in imaging. So in other words, super resolution imaging. And Jennifer was a pioneer of using fluorescence. And that allows us to ultimately look at things live and ultimately also look at individual proteins instead of looking at just groups of them. And Harold is a pioneer in many of those areas, as well as in new techniques of EM that don't require us to cut sections and in fact allow us to get much, much higher resolution. So one of the challenges of all these different methods that we use are there's a lot of acronyms to remember. And fortunately, when you're from the government, you get used to using acronyms where sometimes you can't remember exactly what they stand for, but you use them like you do. And that's sort of what I've done here is there's a lot of different imaging approaches that we use. And a lot of this is still done at Genelia just because these microscopes are not available commercially. They're still, they're homemade, and over time they get commercialized, but we've been very fortunate to have the ability to use many of these, sometimes 5 or 10 years before they become commercialized. And what they do is they allow us to have a significant amount of power in answering questions that with traditional approaches would not have been possible. Now, what this imaging does, I think that's unique, is it allows us to think of, again, getting back to this term about pathology, it allows us to think about pathology in a new way. And that is not just thinking about pathology as something fixed that's in front of you, but as pathology as something that's dynamic. In other words, when we examine patients, we watch them when they're moving. We don't have them sit perfectly still. And we manipulate things, we have them do things. But in most of our biology, we take still images and we publish those, and we never actually look at how the cell is moving. But of course, pathology can affect motion as well as static appearance. And just as an example from one of Jennifer's papers in Nature a few years ago, this is just an image of a cell that's moving. And you'll notice all the organelles are in different colors. This is what's called multispectral imaging. So you can actually look at multiple organelles at the same time. In fact, you can look at all of them at the same time. And that allows you to see different patterns of motion. And we've started using these patterns of motion in our disease studies. Another thing you'll notice is the ER is in yellow, and the ER pretty much just fills, this is just a tissue culture cell, it pretty much fills the entire cell. And one thing that you'll notice about it, it's moving a lot. And you think, well, okay, everything moves, it's there's heat, it's a thermal motion, but it turns out you can shut all this motion down with just inhibitors. So this is actually, the cell is doing this on purpose, and it's exerting a tremendous amount of energy to do this. So the motion is important. And it turns out that if you watch the ER, it ends up, I think, over a 15-minute period from Jennifer's work, feels like 97% of the cell. So it really senses the entire cell, and that gives us clues about its function. When you think about a small cell, that makes sense. It's easy to do. However, if you think about a 1 meter cell, imagine how important it is to make sure that ER is shaped and distributed properly. And we think that's a lot of the basis for our disease, is that in these long neurons, that the right types of ER are not in the right place. And that ultimately leads to dysfunction of the cells. If you just isolate the ER and look at its motion, you see that it moves a lot. This is a technique called GI sim, which is grazing incidence. Let's see if I can remember what sim stands for. Super resolution imaging, basically, at 40 hertz. So this is at a much higher frequency than you'd normally see imaging done. And that also makes an important point that a lot of the structures that we see in live imaging, if you don't image them fast enough, they actually look different. And I'm going to show an example where a really big fundamental error in what we think of as the shape is just because we image too slow. And we don't image at high enough resolution. And you'll see on the left that we have to image over a period of even milliseconds. We can see dramatic movement of this organelle. And it's important to remember that it's not just like these are nice videos and we can watch it moving. This motion is something that's impaired in disease. And if you can't see the motion, you can't understand how the disease manifests. So again, sort of this ability to see things not just at high resolution, but live has been a real advancement. But in addition to just the motion of an organelle and how it's moving, we also want to emphasize seeing things at higher resolution. So there's a couple techniques we use for that. One of them, I'll show you, and there's lots of them. This is called paint. And it's point acquisition and nanoscale topography, I think. So what this is a single molecule approach to get by the diffraction limit of microscopes. And you'll see at the top, this is actually a lattice light sheet image. So this is diffraction limited. And you'll see we'll zoom in on a structure, and then we'll switch over to paint. And you'll see that what looks sort of like a sheet is now clearly not. We can see much more detail of the structure of the membranes of the organelles. And we just this is just staining all the membranes in a cell. You can see there's vesicles, you can see there's ER going everywhere. Lipid droplets are the little white things, I think. So you can see a lot of detail that you can't see with these traditional microscopy approaches like lattice light sheet or traditional confocal microscopy. And you think, well, okay, these are nicer pictures, but is this really important? And it turns out that it is, because some of the structures that we're trying to study actually aren't what they appear. So here's another image, and this is using another technique. So again, when I was looking at the images of Ray Adams, they used to cut the brain sections and they cut EM sections and so on, and then they would look at them under microscopes. Well, we can't do that anymore because there's no way you can cut an EM section thin enough to be able to study the ER. So we use a technique called focused ion beam scanning EM. And what that does, instead of actually cutting a section like with a blade, it essentially polishes it off with a focused ion beam. The great thing about this is that you eventually get these images at like 4 nanometers in the Z, like if you think of it at XYZ, in the Z plane, it's every 4 nanometers instead of, how thin can you cut a section? 40 nanometers or 80 or something like that. So it's like an order of magnitude better than that. And that lets us get a tremendous image of a tremendously detailed image of structures. The downside is it can take weeks to do one of these experiments. An upside from somebody at a place that always worries about space is that once you're done, there's nothing left. So there's no storage issues. All you have is data. There's no block left. You actually end up just polishing it off until it's gone. But you essentially create this massive amount of data that is usually analyzed with machine learning. And just to give you an idea of what the structure looks like, if you look in panel B on the right, the scanning EM image of the ER is on the top. And on the bottom is our calculated confocal image. That's what we'd see under a microscope. So you can see how much detail you lose when you look at traditional imaging versus some of these newer approaches. And just as an example, I put at the bottom is that was what we used to call a sheet. And you see it has holes all through it. It's actually a dense network of tubes. So it's a very different structure. And this becomes important because as we've tried to understand the ER, as we understand the types of structures, we've actually been able to understand how they're made. They're not just, sort of epiphenomenon of messing up a cell or just things that form. Each one of the structures that I'll show you actually has a mechanism that forms it. So it's very important to the cell to do this right. So if you look at this image here, in the upper left-hand corner, this is kind of what we've always thought about the ER, what it looks like. You have these sheet-like structures, you have these tubes connecting them. And the good thing is that's actually correct. We do have, that does exist. But if you start looking through these other images, you'll see things that maybe you don't recognize. Like in B, well, that makes sense. We can also study the ER, not just in isolation, but how it interacts with other structures. Ribosomes are the easiest ones, but all the other organelles as well. Because if you think about it, when I show that image of all of the organelles moving, when you're near the nucleus, everything is very compact, it's very tight. And those interactions are a part of the function of the organelle. So we have to understand those well too. But you'll notice all these structures like 3D matrices in E and 2D matrix and stacked sheets and twisted sheets. And it turns out that over time, there's going to be a series of papers coming out that we're actually starting to understand how these are formed. So if you think about the way we learn about ER as sheets and tubes, that's just really scratching the surface. There's all these other structures that have very specific ways that they're formed. And of course, those are all potential areas where disease could impact them. And I'm going to show an example later where in one of our disease models, we actually found 2 new forms of ER that are subsequently been found in normal cells too. So sometimes our mutant forms or our diseases actually tell us what to look for in normal situations as well. So a lot of what I've showed you is, the pathology of the ER, or not really pathology as much as just the basic structure. Of course, pathology is how that gets impacted in disease. But another aspect of any organelle biology, and again, when we think about diseases, we think mitochondrial disease or lysosomal disease, it's important to remember that all of those organelles interact with others. But our ability to study interactions is very limited because we often fix cells. When you fix a cell, you alter the interaction. So it's almost like you'd have to get lucky to be able to see, a real difference. And I'll give an example of that. If you look at the structure on the left, that's a mitochondria and an ER. And historically, we would measure that contact site, and we would say, okay, if it's more of it or less of it, that it's different. If it's the same, then there's no change in ER mitochondrial contacts. And I'll show you with this paper that's in press now at Nature that that's not true. We can actually show an example where the contact area is exactly the same in the mutants in the disease model, but that the actual contact itself in terms of its function is completely different. And if you look at on the right is what the contacts look like in a cell. And you'll notice that there's the sausage-like things are mitochondria, and the ER is in sort of this translucent kind of green, and then the contacts are in red. And you'll see that the contacts are not that, they can, depending upon where you cut a section, they could look very, very different. So it emphasizes the fact that in the future, I think if we want to study these contacts, we're going to have to use techniques like this. Now, this doesn't even use fixation. It uses high pressure freezing. And to tell you how cold you have to do some of these experiments is that, and a lot of these were using liquid helium, which I think is like 270 degrees below 0 Celsius. So you have to use really, really cold to freeze. So much colder than like liquid nitrogen and things like that. So the HPF is high pressure freezing. Because we found at this level of resolution that fixation created artifacts everywhere. So pretty much everything we do now when we study contacts is we freeze. And again, this was done with Harold Hess's lab at Genelia, as most of the imaging has been done, just because that's really the only place where a lot of these microscopes exist. So again, we see the importance of these contacts, but we also see the importance of figuring out new ways to study them, not just by area, but by potentially the functions that they subserve in the cell. So again, if you get back to this image I showed you before, this is sort of the basics of what we see in terms of of imaging of the ER. But as I show you, when we show images like this, as we often do in our reviews, it emphasizes how we can form a tube, but it doesn't emphasize key things like how it gets to where it's going, or how different types of ER are distributed within cells, and how the ER actually is involved in organizing other organelles throughout the cell. We had another paper in Nature last year that really did show that a lot of the function of ER, and one reason we think it goes all throughout the cell, is it not only has a role in, again, we don't know what it's doing in every part of the cell, but one thing it does do for sure is it distributes the organelle through contacts. So contacts are very important, as well as the proper shaping and ultimately the proper distribution. And all those things we think can be involved in disease. Now, one of the challenges we face, and I showed you this image just before, is that if you think about modeling disease, so again, thinking back to Ray Adams, a lot of the studies, a lot of the science of neurology was on human specimens, biopsies, brain tissue, so on. I think one big difference, as I was thinking about what's different about pathology now, is that we emphasize a lot more models of disease. So again, they're not the same thing, but they're models, and they're models that we can manipulate in different ways that might give us new kinds of insights. So even though they're not, like this mouse that I'm going to show you does not have spastic paraplegia, but it models several features which we think are interesting. But another challenge we face sometimes, and this might not be as true for people that are studying cognition or other types of degenerative diseases, but if you're thinking about a length-dependent disease, we have a huge challenge, is that mice are small and people are big. A mouse is 10 centimeters, and the neurons we're trying to model are 100 centimeters. So how can we do that? And I was frustrated early in my career when we would make these mouse models and they'd be normal. And then, I would speak to mouse experts and they would say, well, of course it should be normal, because if you're really modeling the disease, the mouse shouldn't have any symptoms or they should any phenotype. And I'm like, well, that might be true, but we have to have a phenotype. So what we've decided to do, and this has worked very well, is we've combined different genes that work in the same pathway to get a stronger phenotype. So you'll see here's two female mice at 8 months old. And what you'll notice is that the one at the top is normal and the treadmill is going faster. At the bottom, the treadmill is going much slower, and we've mutated two of the HSP genes that interact with each other. And what that's allowed us to do is get a stronger phenotype that we think is still true to the biology, but allows us to have a strong enough phenotype to intervene and test new therapies. So you'll notice the mouse on the bottom really looks paraplegic in many ways. And the one at the top is walking normal. Now, if we just mutate one gene, we get a far less severe phenotype, and it's too difficult to see if our interventions are working. So to sort of show this quantitatively, if you look, you can really just look at rotor-rod and ledge test, and the bottom and top are just male and female, so they're the same. But if you look at the top on the rotor-rod, you'll notice that all the mutant animals are different than all the wild type. So it's a huge difference. And it's the kind of difference that we can exploit to be able to test new therapies. And one great thing about this is another challenge we face in some of these slowly progressive disorders is that if we have to wait for our models to age 2 years, that slows things down quite a bit. This model is not only clinically robust, but it also, we start to see an abnormality just at 4 weeks. And by 24 weeks, most of the effect is gone. Most of the effect is there. So we have this ability to intervene in a relatively narrow window and hopefully be able to get information on our therapies relatively soon. And when I was thinking about, again, the differences in sort of the Ray Adams era versus now in terms of pathology is that one thing I think in all of our systems emphasize is quantitation, not so much just the description of what we see, but the quantitation. And I think that's because for the great reason that there's so much focus now on using these to test therapies, is that we need to see if our interventions are working, and those are quantitative decisions. Essentially, we need to see quantitatively how much of an improvement we're getting with different types of interventions. So again, I think that's been a great advance. The way we think about pathology now is this use of all these model systems. The model systems also allow us to look at structures and look in ways that we can't in mammalian cells or people specifically. However, I will show one case where we actually can see something. This is a traditional EM. This is like you could do in any kind of tissue, not using any of the fancy technologies that I showed you. And you'll notice that on the left is an axon kind of running up and down. And you can't see the ER because it's just tubes running along with everything else. And you'll see a couple mitochondria because they're much bigger. However, on the right, you notice that all of a sudden that the ER has been shifted. It's no longer going longitudinally, it's going transverse. And this has ended up being an interesting thing because this was one of our first tests of the fib-sem. So if you see what you see on the right, you can see that it's clearly abnormal. The mitochondria are all fragmented, and there's these tubes that are going sideways that aren't supposed to be. But we really wanted to say, well, what if we put a lot of effort into this, what does it actually look like? And that's in this image here. So this is actually what you see with fib-sem. And this is fib-sem done in a mouse, corticospinal tract. So it's exactly the tract that would be affected. And on the left, you can see we sort of as we've gone up the block, we've stripped out a lot of the myelin, and you're just left with ER in yellow and mitochondria in blue or purple. And this is an interesting figure because all of this was generated by a machine. It's all the segmentation, which means identifying the organelles, was all done by a machine. All the data that's shown, and those are all log scales. So some of these are really huge effects, are all generated by machine learning because there's just too much data for a person to handle. But what you'll notice in panel A at the top, when you look to the right and we just isolate one axon, is you notice that the ER goes up and down, just like the axon does. However, in panel B, you notice that you have these ladders now. You have these ER structures that are going transversely, and it's a dramatic difference. And you also see that the mitochondria are fragmented, just like we saw in the traditional EM. And what was interesting is when we first started doing machine learning to do the segmentation, the machine couldn't recognize these transverse segments because they're so much different than what I guess what the machine was expecting. So it took quite a bit of training for it to recognize those structures. Of course, once it recognized them, we were able to generate a tremendous amount of data. But what was interesting to me as we saw this is, first, it's great because we know these mice have an ER abnormality. We now see that this ER abnormality, it makes sense in terms of thinking about shaping of the ER being a mechanism of disease. But what surprised me a bit was that first is this structure is so regular. And second is, even though we're seeing it in a pathologic state here, it's actually not a pathologic structure. And we've started seeing this more and more as we've worked on ER. Because if you look at these images here, you'll notice that there's no ladder-like structures there. But I was just at a meeting last week, and we published this one. This was in axons, these abnormal structures. It turns out that these structures are normal if you put them in dendrites. And what they do is these transverse segments go all the way to the surface of the cell, and they're involved in calcium signaling. So calcium signal propagation along the dendrites. But by seeing them in a pathologic state, people knew how to look for them and are now finding them in other places. So it turns out in this case of this disease, it's not that the structure is pathologic, it's that it's in the wrong place. So again, that sort of gets to this idea that when we have these very highly polarized cells, sometimes pathology is not just an abnormal structure, it's structures in the wrong position or in the wrong place. So that's sort of where things are. But one of the things when we think about, again, pathology and thinking about the way we used to do it, looking at sections and so on, is thinking about the pathology of dynamics. And that's something that's hard to imagine because we're always studying pathologic samples. They're either slices or they're sections in front of us. But there's this ability when we use model systems to study dynamic pathology, abnormalities of movement. And a lot of what we do in this area uses this technology called SPT-PALM. And this is single particle tracking, photoactivated localization microscopy. So this uses fluorescence as a way to study individual proteins. So if you look at the ER here in this image, this is, again, Jennifer pioneered this technology along with Eric and Harold years ago, is that we're able to see these ER proteins moving on the ER, and they move really fast. many times we have to slow these movies down. So the ER is actually moving underneath it. And this is a protein called RAB18. It's just an ER protein. But you can see there's an organization, there's a logic to how it moves. So a lot of what we've been focusing on recently is studying the movement of proteins and how that can be impacted in disease. And it's something, again, I don't think we think of that in traditional pathology, but it's an important aspect because when we think about patients and we do things clinically, we always think about motion. We think about how people move, the character of tremors, the character of a lot of the different types of movement that people have. And we can apply those same principles when we study cells. Now here, so when we were doing this study, we wanted to show how dynamics were affected in a disease. And we wanted to study ER mitochondrial contact sites because they're easy to find and they're the most prevalent ones. Mitochondria are easy to see. In these techniques, you can't label any of the structures with gold or anything. You have to do it visually. The mitochondria are easy because they look so different. And we chose VAP-B because VAP-B is mutated. There's a very specific mutation that links to ALS. So we had a good disease link and we had a protein that we do quite a bit about, and we had an organelle contact we thought we could do. And what we did and again, this was with the Lippincott-Schwartz lab over about the last 5 or 6 years, is we studied VAP-B and we tried to create ways to study individual protein movement in the ER, knowing that we would eventually have to study VAP-B. So a lot of these proteins move in different ways, but there's a lot of challenges to studying it. And I won't get into the math, but when this paper comes out, if you want to see the hardest data analysis you'll ever see, because you're trying to analyze a trajectory of a protein on a structure that's moving in 3 dimensions. And it was the 3rd dimension that turned out to be very, very difficult to study, but it was ultimately solved in terms of analyzing motion of proteins. Because ultimately, as I said, we want to quantitate. We want to understand how things change quantitatively. And that it took years to actually figure out how to do that. These images are not hard to draw or hard to obtain, but it's hard to analyze them. So one of the things that we did early on was we decided to compare just a tail anchor protein, one that would just go throughout the ER and the VAP-B. And we hooked a halo tag. A halo is just a fluorescent tag. It's very bright. It's very easy to see. And if you look at the images on the right, at the tail anchor, when you start looking at the little Roman numeral, the little eye and the 2 eyes, it kind of looks like the ER. It goes everywhere. However, when you look down at VAP-B, you'll notice that it tends to coalesce around a specific area. And this was great to us because one of the things we always wanted to do was to be able to study contacts dynamically. And this sort of gave us the clue that we can do that because you'll notice that the VAP-B is actually not moving everywhere. And the reason it's not moving everywhere is because it's at a contact site. So if you look at images like this on there's this mito and ER, and then these localization densities or how much of a protein is in a given position. And if you just look at the top in the middle on the tail anchor, you don't really see any localization, any specific clustering. However, if you look down in the panel in the middle where there's 4 cubes, if you look on the bottom right one, you'll notice where the arrows are pointing to, as well as where there's this boundary of the mitochondria, you'll see that there's areas where it's coalesced. And most of those coalesced sites actually occur at contact sites. And that's in the far right, you'll notice these areas where it's red and the arrows are, that's actually a contact site. So what was great about this is we could use VAP-B as a way to study contact sites in living cells. And it turned out that when we compared this to the images of the focused ion beam scanning EM, it was the same. So in the same way that we can use EM to map out these contacts structurally, we can actually use the movement of proteins dynamically. It moves differently at the contact. And in a sense, that's what a contact is. Contacts aren't that they're glued together, it's that they influence one another. And part of that influence is on the motion of certain proteins. So of course, this is just one protein and we're doing a lot more. But what was really exciting about this, and again, thinking about this from a pathology point of view, is that when you look at this ALS mutation and you compare it to a normal protein that doesn't have the mutation, you'll notice a dramatic difference. The one at the top is the mutant. The one at the top is wild type, the one at the bottom is mutant. And you'll notice the one at the top is in blue, it's moving fine. And it's just one protein, by the way. And at the bottom, you'll notice it's not moving as much. It's tethered, it's over tethered, it's stuck. So this is an example where we can show that that there's this one mutation alters mobility, and that's a pathologic feature. What doesn't change, believe it or not, is the size of the contact site. It's the same, but you'll notice on the left, that's one track that moves all over. It eventually maps out the entire site. That's just one protein moving over a period of time. On the bottom, on the right, you'll notice that it never goes throughout the whole contact. We have to add a bunch of them together to see the contact. So this is, I think, one of the best examples you'll see of how a individual mutation can give pathology that's pathology of movement rather than pathology of that you can see by EM or any other fixed area. You would always notice that the contacts were the same, but using this method, we can see the contacts aren't. They're different because the proteins themselves move differently. Now, one of the reasons that I've been doing this, and this is just my last couple of slides, is that one of the things that's puzzled me, and I think puzzled many of you that work on different diseases, is the variation when you take one gene and mutate it in different places, how you get such completely different diseases. And often our typical terms of gain of function, loss of function are just not enough to explain that. So we say there's other genes involved, things like that. And I don't think that's always true. I think there are differences that we can't see. And I'm going to give you an example of just like 3 slides, which are different diseases. So they're all affecting this elastin protein, which is the one that fuses the tubes together in the ER. This is a girl, you can see she's walking at a marathon. So she can walk. You can see she's spastic just by the way her feet are, by the way her legs are, and obviously she's using a walker. We can understand everything we need to know about her mutation and what it does. It doesn't affect the GDPase activity very much, but it affects the folding of the protein. So we understand quite a bit. This is probably a toxic gain of function in some way because this is a heterozygous mutation, and this is a dominant disorder. So we understand quite a bit about this. And again, she walks and so on. However, years ago, we worked with another group. And actually, we did this one by linkage analysis. How often do you do that now? And actually, a large family with hereditary sensory neuropathy had a mutation in the same gene, 12 residues away. Didn't seem like it should do that much different. Again, we found it by linkage. It's definitely the pathogenic cause, but this is completely different nerves. This is the sensory nerves, and you can see the sort of the stigmata with auto amputations and sort of things in the feet. We never see this in our HSP patients. One of the patients had a little bit of increased reflexes, but not spasticity like you normally see. So one of the questions is, how can you get these dramatic differences in phenotype on the same gene, same type of mutation, a missense change, 12 residues apart in the same basic area? So, and then to go one step further, and this is another a child with a very severe form of spastic quadriparesis. And, a lot of these kids have extremely severe forms and a lot of other symptoms. Another missense mutation, not that far from the ones that we talked about. And Darius Ibrahimi-Fakari, did an extensive study of some of these, especially focusing on these kids. And it was interesting. So when you look at the SPG3A, this is the same gene, mutation spectrum, all these missense changes everywhere, all of them that cause these severe, or almost all of them that cause these severe forms, are within a couple residues of each other. And it's on the red toward the right. There you'll see 407, 408. And since then, we've gotten a lot more patients, and they're all fitting into that one little area. So I think that tells us that there's more to understanding ER morphology than just this abnormality. We have to really dig deeper and understand how it's changed, in what way. Is it something dynamics that's changed? Is it a specific type of shape change? And I think that becomes important as we start to think about how we localize. So again, when I think about a lot of this, the old pictures you show of Ray Adams and everybody looking at the brain sections and the images, a lot of the localization is like what we're doing on the left. We're looking at, and you can see in the images here, they're looking at parts of the brain and how those parts of the brain ultimately give rise to the symptoms that we see. And that makes sense. However, for a lot of these disorders localize to every cell in the body. These genes localize to every cell in the body. These abnormalities are present everywhere, yet there's only specific types of neurologic dysfunction. So in many ways, we have to understand first is why there's some selectivity. And maybe it's as simple in some cases as just long nerves versus short nerves. But also we have to think about how diseases relate to each other in different ways. Like I showed you an example of if we localize traditionally, these would be in different cell types. You know, a neuropathy versus HSP is not the same cells, but mechanistically it is. So if we think about new ways of doing localization, we would say, well, ER shaping, but that doesn't really do it either, because I've already showed you, we know why ALS patients, what their abnormality is. They have different ER shapes, but we think that's secondary to these abnormal interactions. Whereas in the case of the neuropathies versus HSPs, and again, I have lots of examples other than the one I showed you, but how can this one gene give rise to two completely different phenotypes? So what we're doing is we're actually studying these. We're making mouse models against the individual mutations and also doing a lot of the dynamic single particle tracking studies. And I can tell you, we're starting to see differences in different types of mutations in terms of movement. So I think that, you know, we always thought that, well, okay, the shaping is not just one type of abnormality, it's a lot of different types. And shape is not just what we see statically, the shape is also how the individual proteins are moving and how the structure itself is moving. And all these, I think, will ultimately help us tease apart how one gene can give rise to so many phenotypes. And ultimately that it's not, again, never as simple as just gain of function, loss of function, but in these types, it's gain of different types of function. And I think that will go a long way toward not only helping us understand the disease pathogenesis, but also allow us to, you know, again, target our therapies toward what is really abnormal. So again, I think it's sort of a different way about thinking about localization in clinical neurology, but it really gets back to, you know, Ray Adams and a lot of the pioneers of neurology and neuropathology is we're really doing the same thing. We're as neurologists, or you know, we're neurologists of cells, we're really trying to understand the differences at a very specific level, and that's not only for cells, it's for our model systems. And the more we understand those, the more we understand the detailed pathology that we see. And again, the difference is that I would say now that we have so many model systems, our pathology can focus on other things. It can focus on iPS cells, it can focus on mouse models, fly models, worm models, whatever. And because of the manipulations we can do in those systems, our ability to study different types of pathology is in many ways greater. And again, I've tried to show that a bit with some of the imaging that we can see. And sort of, you know, when we think about what we can do all the way from, you know, tissue pathology all the way to a gene mutation, one of the things I think that's been missing for many years is the ability to, you know, sort of fill in that gap of super resolution, being able to see, like in the case of a genetic disease, how a single protein can have an effect. And what's really remarkable, and we're doing this in one study now, is to the point now where we can even see the structures of these proteins in the cells. So we can see the mutation and how it affects the structure, not in a crystal structure, but actually in the cell itself. Now that's hard to do right now, and we can only do it for certain types of proteins. But I think it really does sort of complete the sort of the line all the way from pathology, you know, to the gene mutation in many ways. And I think that's going to play a big impact on our ability to, you know, start targeting therapies, because we'll really have good readouts in the future. So I just wanted to thank all the people that I've worked with. Again, this is just really my list for mostly this topic. I just wanted to emphasize a few people. Again, my lab is in bold, some people who've done some important studies. Heng-Li Zheng has moved on to a faculty position at Peking University. Johnny Nixon-Abel, who is one of our pioneers in ER morphology, was a Sir Henry Wellcome Fellow and has just received one of the really long, big Wellcome Trust funding to become a faculty member at University of Cambridge. Again, we've benefited tremendously in bold at the bottom with working with Jennifer for about 15 years now, and Chris, who's moving to San Diego to set up his own imaging lab, and Harold. I mean, we couldn't have done any of our studies without the tools and the approaches they have at Howard Hughes at Genelia. And Darius, I'd like to emphasize that he's really helped us in terms of HSP clinical studies across the city with a real big focus on identifying these in children, which was, again, for many, many years, I think, an area that wasn't as well understood. A lot of these kids were mislabeled and misdiagnosed. And I think now there's a lot better effort toward really identifying some of these rare forms of HSP in children as well. So thank you. And I think it looks like I have plenty of time for some questions. Thank you.
Great, thank you. That was really expensive and beautiful. I think, your Dr. Adams, President described pushing technology to, beyond resolution, super resolution. I they you alluded at the end to priority as a technique to look at proteins within the cell itself. Can you give us a little hint as to what we're thinking?
Yeah, so what we've been doing is, so we can, the hardest part about doing that is actually not the resolution. We can get atomic resolutions of these proteins. It's finding them. So, we're actually doing that with a project now, and I hope they don't hate me too much. So again, I guess for those of you that are on the video, just to emphasize, so Brad asked, I don't know if you could hear him, but he asked about, how we use some of these, especially these cryo techniques, these high pressure freezing imaging with the FibSem to see protein structures. And right now we can do proteins at about the 300 to 400 kilodalton range. So we can certainly do, obviously things like ribosomes are easy to see. We have a couple multi-protein complexes we're looking at. So if you were saying, okay, does this protein have you altered stoichiometry or something? We can see that. Whether we can see the actual protein change in structure, probably not yet. But that's one of the things where I think we really have to push a little bit further. That's hard to do right now. But if we had to take, I have a protein called spastin, it can form either hexamers or monomers, and we don't know what it's like. We know what it's like in vitro, but not in cells. We can do that. We just have to find the protein. And that's the challenge, because, it's not a lot. It's not easy to find them. Some of these are not that common, and it can take a long time to find it. But once we find it, we can answer questions at a structural level like that. And I think that could be very valuable too, because again, we can start, it's very helpful, especially, well, the easiest thing to do is, as I showed, we can see where proteins are within the cell. That's easy. But seeing how how a mutation alters, oligomerization or something like that, I think we can do that now, and we're trying it with one of our projects now. We haven't actually succeeded at any of these, but I think it will.
Very good. To second Brad comment after the presentation, fascinating. We're raising up here. Very good. Appreciated your dynamic mythology. But the question we might ask is, in HSP, we're on track with the degenerative, and the linkage between what you're talking about here and the neurodegeneration. I didn't hear too much about it.
You mean about specifically the HSPs in people, the degeneration? Well, that's a good point. So in HSPs, we have two types of abnormalities. We have developmental abnormalities because we have a lot of children that have these. And of course, in some cases, we see a developmental abnormality followed by a degenerative path. And we also see, again, the more classic sort of axonal, maybe dying back degeneration that you see. And there hasn't been that much traditional pathology in the HSPs. And there's a couple reasons. First, and again, these diseases have been around for well over 100 years and described, but they weren't described genetically. So, we don't know exactly what some of those old pathology cases are because there's not a genetic type link to it. Was it really HSP or was it something else? More recently, some of them we've gotten a little bit more, particularly with one SPG 11 and 15, which are ones that are sort of endolysosomal oriented, but also are fatal earlier. But most HSPs have normal lifespans. And since most of these genes have been identified relatively early, we have very little actually in the way of traditional pathology. So it does make it hard. A lot of our pathology that I described is based on what we see in our model systems. It's not based on what we see in people. So that's a big gap. We would love to have more human pathology, of the spinal cord, obviously, but we don't have that right now. And in the cases where we do have it published, it's rarely a typical form. There's a couple that are genetically linked, but they're not typical presentations. And again, I think a lot of the reasons these are diseases that were, for the most part, most of these genes have been identified within the last 10 or 15 years. And most of the forms of HSP that are most interesting to us that are not complex forms that have a whole bunch of other abnormalities, the patients have normal lifespans. So I think that's part of the reason we don't really have that that one piece we are missing is really good human pathology, and that will come over time, but we don't have it like you would for Alzheimer's or stroke, or a lot of the diseases that you focus on.
What about it? Awesome. Patients.
Mhm.
Better. Okay. Well, they're autopsy.
Well, yeah, so what I showed you is we've done traditional autopsies. I didn't show that, but as I showed you in that image of the corticospinal, that reconstructed image, we can see the ER abnormality in that. Now, if your question is how are the axons? Do we see degeneration? Yes, we do. I just didn't show that. So what we can do is with the FibSim, it's great because it's easy to see myelin because it's so dark on EM. So we can actually measure the diameter of all the axons, and we've done that. And obviously, they're much narrower. There's a lot more evidence of degeneration. I just didn't show that because the ER was what I wanted to show. But we have all that information. So with the mice, we always will do a formal, neuropathologic exam, pathology throughout the whole body, actually, to look for anything else that might be abnormal. But I think it does get to the point where these are just model systems, and we can do that. What we really need in the field, or one missing gap, is sort of the traditional pathology, and I think that will come over time. We just don't have it now.
Yeah.
That's great. Far exceeding my knowledge of cell biology. So then you showed a lot of schematic of that ER for some ER proximal axon. How far out does that go? Can you go all the way to the end of the axon? Is there some way to extrapolate from that why you see so much problem with long axon systems, but not the perfect?
That's a very good question. Yeah, well, again, in peripheral, we do see some. It's just, again, some of the sensory symptoms. It's just that people don't complain as much about it. And I should have emphasized, in the ones I've showed you at Blackstone, there's 3 forms, and there's one form. There's different forms in different cell types. So some of the specificity is just where it happens to be. But it's an important question about the ER distally. So I should have emphasized this. 1 meter axon, your ER goes all the way down the end. And in fact, when you look with EM, pretty much even any tiny axonal process, even the tiny little 100 nanometers or whatever ones, will have an ER tubule down the middle. It literally is everywhere there's cytoplasm, there's ER, and ER is connected everywhere. So, and what I didn't show you is that those abnormalities, those ladder-like structures, are present more distally than proximally. So they are where you would expect the disease pathology to be. We also see other structures of ER that I didn't talk about today that we've now understood how they're formed. They're also more prevalent in different parts of the cell. What's interesting is like at a growth cone where you have, or at a cell process where you have it as expanding, there's these clusters of ER, and it's probably so that as the cell expands, it can fill it with ER immediately. That tells you how important it is. But they literally have these clumps of ER ready to distribute into these forming processes. I didn't show that, but we have that data. So again, I think it emphasizes that for whatever the ER is doing, it has to be everywhere in the cell. And if you imagine if you disrupt the morphology or the distribution, which almost a lot of the mutations do, a lot of those will ultimately result in ER not getting to where it needs to be, or the wrong type of ER. And that's what we think link dependency is. In a small cell, it's easy to do. In a 1 meter cell, it takes a lot of effort. And that's where we see the vulnerability. And these shape changes alter that ability. And it ends up being a huge effect, like I showed you with the axons. The ER was in the axon instead of the dendrite. It was completely mislocalized. Whereas in a smaller cell, you might not notice that. That is sure. Brian?
Right. I'm particularly intrigued with the hierarchical dynamics. So the focus on single genes that have different phenotypes and localization, defect, and but we also see kind of stereotypes that are very similar to the way so defects be one others too, which is B and there's the less question. Here's the chemical signature associated with this. And that's the how are you given the importance of see specific chemical signatures, the biomarkers, or that would correlate with these specific structural events that you're
We are seeing some, yeah, and what, again, I didn't want to get into a lot of the complexity, but it turns out that some of these, some of these are not cell autonomous, as you might imagine. Some of these mutations are actually, it seems like they're affecting the astrocytes more than they are, and that the damage to the axon is secondary to that. We see abnormalities in several things that we've been able to treat in some of our model systems related to cholesterol efflux, by targeting liver X receptor, the liver X receptors, and we've been able to restore a lot of function that way. But I think one of the points and one of the things that we're going to try to do in the longer term is exactly what you're suggesting is, what types of biomarkers can we look at? So a lot of people are doing lipidomics and so on to see if there's alterations there. A lot of people are looking at now, especially because of these ladders, calcium signaling and how that's impacted. So I think that that's what we ultimately had to do is we spent a lot of time understanding morphology, but ultimately, how does that lead to a marker that we can measure? Rather than doing these tests that we do are not something that you can follow, obviously, clinically. So we want to translate that into something that we can follow clinically. And a lot of that is now based on, given the importance of the ER and a lot of lipid metabolism, a lot of what we've been doing, and we had some preliminary data when I was at NIH in terms of even some lipid abnormalities or changes in serum. So I think that's the direction we're moving. Again, we spent a lot of time just imaging, but obviously that's an important gap that I didn't mention as much. And again, when we think about the old days of doing pathology versus clinical, often they didn't look at biomarkers, but we recognize how absolutely critical that's going to be for a rare disease. And that is a lot of what we're doing now, both in the iPSCs and ultimately, and in the mice themselves, we do a very detailed like serum analysis and CSF analysis. We have some new mouse models that we're doing that for exactly what you're suggesting is, can we identify markers that we can follow longer term? One thing I could mention is that one abnormality we saw in the mouse I showed that I didn't show in the talk was a difference in phosphorylation of neurofilaments. So the levels were fine, but the phosphorylation state was dramatically different. So I think it is important for us to notice those things and think about those as we think about our markers, because it might not just be the total levels, it might be a modification or something like that.
Very interesting.
Thanks. Sure.
Really soon.
Thank you. Oh, that's amazing. Oh, thanks. Fun stuff. Huh? Yeah, absolutely. We're going to get this gene editing stuff on post. We're doing that now. We're doing the database stuff. We're doing that, and I'm about to do this. So we're going to see if we can actually not only correct the phenotype, but correct that. So we're doing that now. The Dave was interested in doing that with us. So no, we're doing it all at Jackson Labs too, because they're doing a lot of the gene editing with Dave at Jackson Labs. So we're just having them do all the housework so that also